Trypanosomes. Trypanosomes. Trypanosome. Phylum Sarcomastigophora. Subphylum Mastigophora. Mastigode is a Greek word which means flagellum. Members are flagellated class Mastigophora, order Kinetoplastida. Member have a kinetoplast, a small organelle contain DNA present in the part of mitochondria at the posterior portion of the body just behind the blepharoplast. Family Trypanosomatidae. Family Trypanosomatidae. Genus Trypanosoma and Leishmania. Members are hemoflagellated, leaf-like or may be rounded in shape. During the life cycle for developmental stage may occurs trypanomastigode stage, trypanosome stage, kinetoplast present posterior to the nucleus near posterior and an undulating membrane well developed. It found in both vertebrate and arthropods. Epimastigode stage, Cryphidial stage, kinetoplast and axonema are present anteriorly to the nucleus and undulating membrane, short and found principally in arthropod. Family Trypanosomatidae. Family Trypanosomatidae. Promastigode stage, leptomonad stage, kinetoplast present at the anterior tip of the body, UV absent and found in arthropods or plant. Fourth, amastigode stage, Leishmania stage, rounded kinoplast present at high angle or to the nucleus and found in vertebrate and arthropods. Out of 4 developmental stages, only 1-2 stages are found in the life cycle of parasite. Family Trypanosomatidae. Different stage of development. Trypanostigode, epimastigode, promastigode, amastigode. Genus Trypanosoma. Genus Trypanosoma, mostly extracellular or intracellular parasite found in blood and tissue fluid except Trypanosoma cruzi, which is a intracellular parasite occur within the cell of reticuloendothelial system and in the cardiac muscle. Trypanosoma species are transmitted from one vertebrate host to another either cyclical or mechanical through blood-sucking arthropod except Trypanosoma equipedum, which is transmitted during sexual coitus. Sexual process absent and trypanosome multiply by binary or multiply fission. Genus Trypanosoma morphology. Typically, trypanosomes are elongated leaf-like, single vascular type nucleus at the center of the body. Single flagellum originated from the blepharoplast at the posterior end of the body. DNA containing elongated organelle known as kinetoplast present at the just posterior to blepharoplast. Flagellum attached to the body of trypanosome form undulating membrane. Genus Trypanosoma transmission. Transmitted by blood-sucking arthropod, usually an insect either cyclically or mechanically depending upon the development of parasite. Cyclical transmission through the vector in which trypanosome multiply. 2 types according to developmental site inside the vector. Anterior gestation development, salivaria. Epimastigode form develop into infective metacyclic form with a glycoprotein surface coat infective form in the foregut of vector and form inoculated into vertebrate host during blood sucking of vector. Salivaria found in T. congolense, T. brucei, T. rhodensiense, T. gambiense, T. vivax. Genus Trypanosoma. Posterior gestation development, stercoraria. Multiplication occurs in the midgut of insect and metacyclic trypanomastigode form are passed in phases of arthropods. Infection of vertebrate host occurs by the contamination of the skin, example T. cruzi, T. theleria, and T. melophagnum. Trypanosoma mechanical transmission. No developmental cycle occur in the vector, and trypanosome survive for a short period in the proboscis of vector. Trypanosoma evansi is transmitted by interrupted feeding habit of Thebanus Stomachsis flies. Trypanosoma equipadum also mechanically transmitted by sexual coitus. Trypanosoma equipadum also mechanically transmitted by sexual coitus. List of trypanosoma. List of trypanosome. List of trypanosome species, final host, vector, disease, symptom. Trypanosoma evansi, horse, dog, camel, cattle, dog, sheep, goat, wild, lab, animal. Tabenas, chiefly stomachsis and leprosia, sura or tabersa in camel. Trypanosoma equine, Tabenas species, male decadres, weakness of hind quarters, leading to staggering gait. Trypanosoma equipadum transmitted mechanically by coitus equine. Venerable disease called durine and lesions known as dollar spot. Dollar spot, Trypanosoma equipadum. Trypanosoma theleria. Trypanosoma theleria, Thebanid fly, cattle, non-pathogenic. Species, vector, final host, disease. Trypanosoma gambians, sissy fly, glossina species, man, cattle, sheep, goat, horse, dog, and cat. Gambian sleeping sickness in West Coast Africa. Trypanosoma rhodiense, sissy fly, glossina species, man, cattle, sheep, goat, horse, dog, cat, and laboratory animal. East African sleeping sickness, Zambia, Zimbabwe, Sudan, Tanzania, and Botswana. Trypanosoma cruzi, redwood bug, Tritomo species, kissing bug, mostly children and dog. Saga disease in South America, Odematous fishes. Trypanosoma congolens, sissy fly, glossina species, cattle, sheep, goat, hot dog, and cat. Nagana disease in cattle. Genus Trypanosoma venereal transmission. Venereal transmission through coitus, example Trypanosoma equipadum. Trypanosoma theleria, cattle. Trypanosoma melophaginum, sheep. T. levici, rat. T. avium, bird, and non-pathogenic trypanosomes. Kinetoplast is absent in Trypanosoma equinum. Trypanosoma melophagium is transmitted by Melophagus ovinus sheep cat. Genus Trypanosoma. Trypanosoma theleria is largest, 60 to 70 micrometer in length. Trypanosome. Smallest of the African trypanosome species is T. congolens, 19 to 18 micrometer in length. Vector of trypanosome levici rat is Keratophilus fasciatus rat flea, and many causes death in nursling rat. Ndama is humpless breed of West African cattle showed trypanotolerance, resistant against trypanosoma species. Trypanosomes were among the first parasitic protozoa to be cultivated in vitro. Trypanosoma evansi. Trypanosoma evansi was the first pathogenic trypanosome isolated from infected equidae in the Dera Lesmel Khan district of Punjab in 1880 by British veterinarian Griffith Evans. Trypanosoma evansi is an emerging zoonotic parasite, and first case was reported from India in 26 September 2004 from Chandrapur district of Maharashtra. Trypanosoma evansi location. It is an extracellular or intracellular flagellate protozoa found in blood plasma and lymph. Host. Urigenius hematoprotozoan parasite found in camel, hot dog, cattle, buffalo, goat, pig, dog, wild, and laboratory animal. Most every disease occurs in camel, horse, and dog, but camel are highly susceptible to trypanosomosis. Trypanosoma evansi morphology generally monomorphic but sometimes pleomorphic 50 to 34 micrometer in length. Single vesicular nucleus in the center. Kinetoplast is subterminal, present posterior to nucleus. Undulating membrane, well developed and has a long free flagellum. Trypanosoma evansi transmission. It is mechanically transmitted by interrupted feeding of bitting fly, example Tapenes mainly, Stomachsis and Leprosia species. Successful transmission can occur if the transfer of the parasite take place within few minutes after bite because parasites do not survive in the proboscis of the fly for more than 10 to 50 minutes. Dog may get the infection by ingestion of tissue from infected fresh meat or carcasses. Trypanosoma evansi transmission multiply by longitudinal binary fission in the blood of host. During multiplication, trypanosomes change their variant surface glycoprotein VSG surface coat and form trypanosome with variant surface antigenic type. T1C does not multiply in the vector and transmitted within short period due to interrupted feeding habit of their vector. Trypanosoma evansi it caused disease is known as sura means rotten. Chronic form of disease persists about 3 years in camel and hence in India it is locally called tabersa. Office International Die Epizootic OIE mentions that trypanosomiasis disease underlies B disease of significance in pathogenic of trypanosoma evansi. Pathogenesis it causes disease called trypanosomiasis or Shura. Progressive anemia may be due to hemolysis released by the parasite which lead to hemolysis of RBC. Increased erythrophagocytosis, hemodilution due to increased production of immunoglobulin, inhibition of erythropoiesis, pathogenesis of trypanosoma evansi, hypoglycemia due to malfunction of adrenal pancreas and thyroid gland, the metabolism of carbohydrate hampered. Trypanosome also consume large quantity of blood sugar glucose and has been estimated as 0.9 mg per 100 million trypanosome evansi per hour which lead to hypoglycemia. Pathogenesis of trypanosoma evansi. C. Undulating fever change of glycoprotein surface coat due to production of antibody which lead to lesser number of trypanosomes in host. This undulating form of parasitemia result in the symptoms of undulating fever. Pathogenesis of trypanosoma evansi, D cell degeneration and inflammatory cell infiltration. It may occur in myocardium, skeletal muscle which result separation and degeneration of muscle fiber, edema and emaciation. Insura, progressive anemia, intravascular coagulation and asphyxia, hypoglycemia are the cause of infected animal death. Trypanosoma evansi is caused most several disease in camel, horse and dog. Symptoms in horse: progressive anemia, urticaria, plaque on neck and flanks, intermittent fever, edema of leg and lower part of the body, conjunctivitis, emaciation, edema of eyelids, etc. Clinical sign of Sura in horse: intermittent fever, anorexia, anemia, edema of the limb and dependent region, dehydration, leathery, weight loss, urticarial plaque in neck and flank region, abortion and incoordination followed by paralysis of the hind limb. Trypanosoma evansi symptom in camel. Chronic form of disease persists about 3 years in camel and hence in India it is locally called tibersa. Symptoms include weakness, emaciation, anemia, intermittent fever, edema of leg and dependent part of body, abortion, disappearing of hum, etc. Trypanosoma evansi in dog. It causes fever, anorexia, bilateral corneal opacity, weakness, staggering gait, and edema of larynx lead to change of voice similar to those found in rabies. Trypanosoma evansi, cattle and buffalo. Per acute, subacute, and chronic form of disease occurs in cattle and buffalo. In cattle and buffalo, intermittent fever, emaciation, weakness, conjunctivitis, edema of leg and lower part of body, circling movement, head pressing against head object, paralysis of head quarter, micturition, etc. Trypanosoma evansi, sura mostly occurs subclinically in cattle and buffalo. These are considered to be the main reservoir of the infection for equine. Stress of any kind, transportation, malnutrition, concurrent infection, and vaccination of rinderpest and FMD may be responsible for per acute and acute form of the disease. Sura generally occurs in buffalo in comparison to cattle. Epidemiology of Trypanosoma evansi. Worldwide infection is reported in India. Sura occurs mostly in post-rainy season, August to October, when the fly breeding is peak and lowest incident in April-May. Diagnosis of Trypanosoma evansi on the basis of history of prevalence of vectors and heart record etc. Microscopic examination, blood samples should be collected from the suspected animal at the pyrexia. Various methods like first, wet blood smear examination for diagnosis of living trypanosome. Diagnosis of Trypanosoma evansi. Blood samples should be collected in Alzheimer's solution if diagnosis to be delayed. Alzheimer's solution composition: dextrose 2.05%, sodium citrate 0.8%, citric acid 0.055%, sodium chloride 0.242%. Diagnosis of Trypanosoma evansi. Dry blood smear examination, both thin and thick in light infection. Smear are prepared and stained with Zema or Leishman stain to detect trypanosome. Quantitative Buffy coat QBC method. Trypanosome are detected on the surface of Buffy coat or WBC layer. Diagnosis of Trypanosoma evansi. Animal inoculation method. 0.5 ml suspected blood inoculated intraperitoneal in laboratory animal like albino mice to diagnosis of latent or subclinical infection of trypanosomes. In positive case, the trypanosome will be found up to 3 days post inoculation in the blood of albino mice. Diagnosis of Trypanosoma evansi. Chemical test. Still Stilbamidine test. In this method, 0.3 solution of stilbamidine isothiocyanate chemical is mixed with 1 drop of serum of suspected animal. In positive cases, an opalescence of precipitate will be appear in 1-2 minutes. It is used for bovine only. Mercury chloride test. 1 drop serum of suspected animal is mixed with 1 ml of mercury chloride solution. In positive infection, a white precipitate will be appeared in a few seconds. It is reliable of camel only. Diagnosis of Trypanosoma evansi. Serological test. Complex fixation test, card agglutination test, ELISA, fluorescent antibody test, etc. are used for the detection of trypanosomes. Molecular test, polymerase chain reaction (PCR) is nowadays more reliable and sensitive test. Treatment of Trypanosoma evansi. Combination of quinapyramine chloride 1 gram, prophylactic plus Q-sulfate 1.5 gram, curative 0.025 ml per kg. Body weight. Through subcutaneous is drug of choice. A single injection gives protection up to 70 days. Diamazine aceturate. Diamazine aceturate. It is used as curative 3.5 mg per kg body weight, subcutaneous or IM or IV. Treatment of Trypanosoma evansi. Isometamedium chloride 0.5 curative 1 mg per kg prophylactic body weight deep IM. Quinapyramine chloride and isometridium chloride work as chemoprophylaxis. Supportive therapy. Administration of 25% dextrose solution is recommended as a supportive therapy because trypanosomes produce hypoglycemia. Control of trypanosoma evansi by using chemoprophylaxis drug like isometamidine chloride and quinapyramine chloride in endemic area before onset of rainy season. By controlling vector like Travenous fly, etc. can be controlled by destroying their breeding ground using insecticide in and around animal shed, proper disposal of feces, etc. By developing genetically resistant breed like N. Dama cattle, which is a trypano-tolerant breed. Trypanosoma equipadum. Final host, horse and donkey. Transmission, it is transmitted mechanically by coitus. Trypanosoma equipadum disease. It caused one real disease called durine. Durine term is derived from Arabic word which means unclean. Disease is prevalent in Africa, America, Russia, and in the part of Asia. Durine disease is now eradicated from India. Trypanosoma equipadum disease occurs in three phases. First phase, stage of oedema, swelling and oedema of penis, scrotum, prepuce, and surrounding skin in stellion. In male, profuse fluid discharge is detected. Second phase, urticarial phase, develop edematous plaques up to 5 cm in diameter under the skin known as dollar spot because it appears as a silver dollar has inserted under the skin. Trypanosoma equipadum 3 phase state of paralysis. It is characterized by anemia, emaciation, weakness in coordination, paralysis, and recumbency followed by death. Diagnosis on the basis of symptoms, urticarial swelling, and detection of organisms in the fluid of genitalia. Trypanosoma equipadum treatment and control. Diminazine, diminazine aceturate, berenil, or antricides. Slaughtering of infected horse. Trypanosoma equipadum horse act as final host and Travenous species act as vector. It causes male day cadres, weakness of hindquarter in horse. Symptoms include staggering gait, recumbency, conjunctivitis, and oedema in eyelids. Special character in trypanosomes. Trypanosomes have antigenic variation due to variant surface glycoprotein VSG. The surface coat of trypanosomes is a 12 to 50 mm thick layer made up of about 10 to the power 7 identical molecules of approximately 60 kDa, which are closely packed to cover the entire surface. These molecules are variant surface glycoprotein VSGS, changing antigenic characters of trypanosomes in vertebrate host. The development of an effective vaccine against trypanosomes is difficult. Thank you.